It's a real privilege and honor to introduce Dr. Kevin Staley to our BRI seminar series. Dr. Staley obtained his bachelor's degree in physics from Loyola Marymount. Actually, Kevin is a native of the San Fernando Valley, like some of us. And so he's a proud Valley boy like me. And he then went on to obtain his MD from UCSD and continued his training at UCSD, doing his pediatric neurology training, and went on to do postdoctoral research with our own Ishvan Modi and David Prince at that time at Stanford. And he was really one of the first to study the role of depolarizing GABA in the hippocampus and its role in seizures as well as in neonatal seizures. Since then, he's complemented the work that he's done using 2-photon imaging techniques to assay chloride concentrations and in different pathological states in different neuronal compartments. He went on to the University of Colorado as assistant professor and over the years made his way to full professorship, moving to Mass General and Harvard Medical School, where he's been chief of pediatric neurology. Much of his work there has been focused on finding for modulators of chloride transporters, which can be used clinically as anticonvulsants and anti-epileptogenic agents, especially in neonatal seizures. One such agent is bumetanide, the diuretic, and his group has pushed this work all the way from preclinical models into clinical trials and has shown efficacy. He's made a lot of basic contributions to the understanding of chloride conductances and chloride concentrations in cells. He was one of the first to discover the importance of impermeant polyanions, which can compartmentalize the equilibrium potential for GABA, even into very small microdomains, even a single GABA receptor level. Finally, his lab has made tremendous contributions in using culture models for studying ictogenesis and epileptogenesis, more recently incorporating chronic imaging and optogenetics, finding key cell types which can start seizures and stop seizures and epileptiform bursts. He's won many honors, including the AES Basic Science Research Award, which is one of the highest awards in epilepsy. And he's been the chair of the NINDS Board of Scientific Counselors and has won a Javits and really a very long list of others. He's a mentor to a large number of physician scientists who've gone on to have their own careers. So I'd like to introduce Kevin. Welcome.
Thanks, Peyman. Guess I can take my mask off now. It's great to be here. I was going to say that I grew up in LA, but Peyman was a little bit more accurate. I grew up in the San Fernando Valley. But it's a great honor to be here because throughout my childhood and early adulthood, UCLA was the medical center, and that's been hardwired into my brain. And years of East Coast pomp and circumstance can't really change that. So very nice to be here. Nice to be invited by an investigator like Peyman, and nice to see old friends, Richard Olson and Carolyn Hauser, who was my first study section chair and still really the yardstick I use to measure the performance of every subsequent study section chair I've worked with, including myself. So then, as Peyman may have mentioned, Ishvan Modi was my first research mentor. So it's a little bit of a homecoming for me to be here. Plumbing problems are the worst, right? There's nothing that ruins a nice evening more completely than finding water someplace it's not supposed to be. And the hypothesis we're going to think about today is that plumbing problems, misplaced water and salt, underlie a lot of the most serious morbidities that we encounter in the ICU, including acute seizures after brain injury, uncontrolled cytotoxic edema, and also residual like chronic post-traumatic epilepsy. So is this going to work?
Wait, what's that picture?
Pardon me?
What's the picture?
Oh, I'm sorry. That's a rusty pipe with all kinds of water spraying out of it. Maybe that's too much of a zoom. I don't know. There we go. So it's something I just got off the internet, of course. So I don't have any financial disclosures. I have a lot of postdocs who have spent years, some of them decades, some of them multiple decades working on this. And without this work, talk would already be over because they've done all the important experiments. A ton of topics I'd like to tell you about today. We're going to do at least the first two light blue ones. And if we get farther, if we have extra time, we'll do some more. Any of these are fair game. So if there's something you want to hear about, just let me know. Because we do a lot of work with acute brain injury and the ion shifts that occur in that situation, we've come across a few findings that are relevant to this neuroprotection crisis that we've been experiencing over the last few years. And so I want to start with a prologue, another look at neuroprotection. And these first two slides are going to hurt a little bit if you're into neuroprotection, because we're going to compare neurogenetics to progress in neuroprotection. And neurogenetics is, it's a golden age. We've had spectacular successes, right? Spinal muscular atrophy went from the worst disease you can imagine, progressive paralysis and death of infants, to something that's disappeared, and now it's on the newborn screen. In fact, I asked my junior residents, after 4 years of training, have they ever seen a case of SMA, and they hadn't. So it's really disappeared from the landscape. So tremendous success. Same with adrenal leukodystrophy, not quite as treatable, but certainly treatable enough to be on the newborn screen. So another horrible disease that's disappeared. Okay, so there'll be a few jerks and pauses because this isn't quite the the most recent version of this talk, but it's okay. It's a good version. So we've tried in the ICU to make progress, and we have in some areas, right? There's reperfusion where, for thrombolytic therapy, we've done pretty well. The number needed to treat is 4, so about a 25% improvement in outcomes. Same with thrombectomy, about the number needed to treat is 4. Not many people turn out to be candidates, particularly in the community. Only about 7% of community stroke has been treated. So if you combine that with the number needed to treat, about 2% are getting better. But, it's a progress. We would like for the other 98% to be able to do something about neuroprotection or about seizures or about cerebral edema. But we don't have a lot of things in our armamentarium. So we have neonatal hypothermia, but we don't have a lot of neuroprotective strategies outside that very narrow age range. Seizures, we basically don't have anything. Acute, there was just a very nice paper in the New England Journal this year showing that treatment with anesthetics of seizures in adults with acute hypoxic ischemic encephalopathy didn't change the outcome very much, not statistically. We'll look at that a little bit more in a minute. And cerebral edema, all we can do is make room for swollen brain. So we can use osmotic therapy, which shrinks the normal brain, or we can shrink the ventricles with a drain or a craniectomy or resection, but we can't do very much for cerebral edema. So these are the things we'd like to do. Can't do too much. Let's talk about these, starting with neuroprotection. So neuroprotection is not an easy literature to follow. Let me just give you my perspective. So in the early 80s, when I was in medical school, tremendous excitement about the fact that neurons take a while to die. So after a just lethal injury, say 5 minutes of arterial occlusion, neurons would still be firing for about 24 hours, still fire action potentials. They wouldn't make any new proteins, but the mitochondria were still working, so still producing ATP, oxidative phosphorylation. They would die over 3 days. Now, at that time, Olney had shown that glutamate was neurotoxic. And from those two observations came this second-hand hypothesis that glutamate was neurotoxic to neurons, and glutamate released by injured neurons and astrocytes was killing neurons over a few days. And then if we could block glutamate receptors, we could prevent this secondary injury. And this paper by Dennis Troy is one of the most highly cited papers in neuroscience, thousands and thousands of citations. So tremendous excitement, sort of a gold rush mentality almost. Over the next 10 years, we learned a lot about neuronal death. We're able to differentiate necrosis from apoptosis. So necrosis is immediate, there's complete ATP failure. swelling of the cytoplasm, rupture of the cytoplasmic membrane. So there's not a lot to see. It happens immediately before people come to medical attention. So it really hasn't been studied nearly as extensively as apoptosis. And, you know, it's not easy to find something that's popped and not there anymore. So we look with diffusion-weighted imaging or leakage of enzymes from the cytoplasm like lactate dehydrogenase. Apoptosis, on the other hand, is slower. It's regulated neuronal death. The neurons shrink, including the nucleus, becomes pycnotic, the chromatin clumps. There's loss of synthetic machinery. So the ribosomes are the first thing that are disassembled by the proteolytic cascade that triggers apoptosis. Mitochondria continue to respire, and the membrane leaks, but it hangs together. And so the recognition was that delayed neuronal death was really apoptotic neuronal death. And we use morphology to some extent to diagnose this, you know, the shrinkage of the nuclei. Caspase stain, so caspase is activated early, and fluorescent inhibitor of caspase activation, or FLICA, is often used as a marker of caspase 3 activation, an early activator in that sequence. DNA fragmentation, polar dyes, so silver, propydium iodide, thallium bromide, and fluoride jade series are all used. They cross the membrane when the membrane is deteriorated significantly, so they can be used to diagnose or to find apoptotic neurons. From 2000 to the present, a bunch of negative clinical protection trials. Rigor has been emphasized. You know, we've found a bunch of novel neuronal death pathways, and we've widened the targets for neuroprotection, still using the same diagnostic techniques that I mentioned above. So why have we struggled so much with neuroprotection? You know, the good news is that we've 1,000 positive preclinical neuroprotective studies, and that was in 2006. The bad news, of course, was about 10 years ago. We recognized that only about 20 to 25% of the actionable small molecule reports that are of interest to pharma companies were reproducible. That triggered a whole sequence of you know, blaming ourselves, I think, for sloppy science and increases in rigor. And now, not only can you not submit an R01 application to the NIH without talking about rigor, you can't even submit a progress report without telling them what you're doing with rigor. So people have really worked hard on that. But think about this from the other side. So it is true that, you know, 75% of studies may not be fully reproducible, but that means that 20 to 25% are. And so if you had 1,000 positive trials and 20% of them are reproducible, then there should be 200 plus robust, reproducible, translatable trials. But when Howell looked in 2006, 0 of 100 clinical trials have been positive, and that's still the case for neuroprotection. So the question is why? And then we can think about a null hypothesis that neurogeneticists who are making all this tremendous progress are better scientists than everybody else. Maybe they're more rigorous than neuroprotection researchers. You know, I strongly doubt that that's the case. And what I would like to say is that the biology of brain injury and the way we diagnose brain injury and follow neuroprotection and neurotoxicity is way more complex than we anticipated. And we don't really understand a lot of what we are measuring. And an example I'm going to give you today, that the intervention, the putatively neuroprotective intervention, can affect the assay of neuroprotection. So I'm going to use the hippocampal slice culture model as a model of perinatal brain injury. So we take P7 mice, they get decapitated, and the hippocampus is dissected out. The neurons near the cut surfaces are injured. It's pretty easy to show that if we take a amalgam of yellow fluorescent protein and cyan fluorescent protein called chlamylion. The yellow protein is sensitive to chloride, it gets quenched, and the cyan doesn't. So we can take this ratio of yellow cyan fluorescence as a measure of chloride concentration. And here you can see that this top, see if I can get a pointer going here. This top area where the cut was, the neurons are swollen and they have a high chloride concentration. So they've been filled with salt, and the chloride moiety of salt is quenching the yellow fluorescent protein. We can take this slice, this acute slice, and culture it for days and weeks on end. This is an organotypic slice culture. And these slices, the surviving neurons synapse onto each other with causing a lot of positive feedback and seizures. And so this is a raster diagram. This is 5 weeks of 1 hour raster diagrams of spiking from the culture stacked on top of each other. We can see initially there's individual action potentials, and later frank seizure activity develops. And the seizure activity is sensitive to dilantin. So if we put that on, we just get interictal spikes and we wash it off, the seizures come back. So a nice model of epilepsy developing after brain injury. So we're going to use that model to look at the consequences of necrosis and apoptosis. And neuronal necrosis is a network disaster. We had studied it in that paper I referenced earlier with the slicing to show that slicing induced neuronal death. So traumatic neuronal death causes a lot of swelling and rupture of neurons. But it's not easy really to study the slice. There's too many neurons dying, a lot of stuff going on. So we use this the tremendous XYZ focus of a 2-photon microscope to really concentrate all the laser power in, say, a cubic micron or so of space right in the cytoplasm of a single neuron, and use that, use any fluorescent dye, we use chloride to absorb that radiation and heat up that particular neuron and rupture it. And this is what that looks like. And we're going to target this cell right here with a little halo around it. And then this is over a course of an hour. It was a 20-minute baseline. And then we rupture the cell, and you can see 3 things happen. So first, the cells become orange, so the chloride concentration is going up. The yellow is being quenched. Second thing is the area swells, so you can see the tissue in that area is distorted as the cells take up chloride, salt, and water and swell up. The last thing that happens is that the neurons turn black, so they actually stop fluorescing, and I'll show you in a couple of slides that that's a sign that the neurons have died. These are the neurons around the target cell that would be right here in the middle, and you can see how the chloride concentration has gone up. And if we look at some sample slides from here, and we stain at the end with FLICA, this caspase indicator, we can see that there's increased chloride cell death, and that's shown not only by loss of fluorescent protein, but also by caspase activation. So a single cell dying causes a tremendous wave of cell injury around it. Now, this isn't due to heating. If we turn the laser power down just 15%, there's no rupture of the neuronal membrane of the target cell, and there's absolutely no damage to the other cells. We can talk about the details in a minute if anybody's interested. So if we compare that to neuronal apoptosis, which I would call planned retirement of neurons. So if I'm going to retire, let's use this as a model. I talk about it with my chair. I take some irreversible steps. I stop applying for grants. I stop taking students, preserve my lab and office. And then we graduate the students out of the lab, publish the papers, recruit and train our successor. And the final step is a retirement party. If we compare that to apoptosis, then a neuron, for whatever reason, makes a decision to apoptose, takes some irreversible steps. So it activates caspases and other proteases, and the very first thing that happens is that ribosomes are disassembled. And that's an irreversible step. So there's no way to make proteins again once this has happened. It's very early in the course of apoptosis. Preserve the mitochondria, preserve ATP, preserve membrane ion transport and channels. And by preserving the membrane components, you prevent the membrane rupture that happens with necrosis, and you prevent cytotoxic leakage of lysosomal enzymes. Network connections are removed right away, dendrites and axons, which prevents network dysfunction. There's a progressive increase in membrane permeability, which attract microglia, could drive seizures in bad circumstances. And the final step is a rapid microglial engulfment. I put rapid in quotes because we'll see that it's not always so rapid. And can be rate limiting when a bunch of neurons die at the same time. So what we can see are the morphological changes we talked about earlier here. So shrinkage of the nucleus and clumping of the chromatin. What's used more, though, is because it's easier to do, is polar dyes permeate the leaky membrane. So, obviously charged molecules like silver, propydium iodide of the fluoride jade series, which are anionic. And finally, the dying neurons disappear when they're engulfed. So with that background, I'm going to show you a very straightforward neuroprotection study that we did in 2012, but other people have done also. So we took the organotypic slice and we just stained for dying neurons. We did a propydium iodide stain, and we found a bunch of dying neurons in the control slices that had been seizing like crazy, and in slices that had been incubated either with TTX, which prevented seizures by blocking sodium channels, or kynurenate, which block at 3 millimolar, which blocks every glutamate ionotropic channel. We didn't get any cell death, and it was a huge effect. So if we look up here, we can see increasing propydium iodide positivity in control slices and none, basically, in kynurenate treated slices. And that happened in the CA1, and the same thing happened in the dentate. So this was multiple species. It was blinded. There was random allocation. There were large effect sizes. There were multiple agents, multiple replications. So a really robust, we didn't do age, sex, and comorbidities, but it didn't really seem like those things mattered here. Very, very robust finding. This is something that everybody would call a rigorous finding, and yet I'm going to show you that it was completely wrong. We used another assay, longitudinal tracking of transgenic fluorescent protein emission, which had been developed by Steve Finkbeiner at UCSF, to look at degenerative diseases like Huntington's, and we applied it to these organotypic slice preparations. So the idea is a neuron that expresses a fluorescent protein, that you expressed either transgenically or using a viral vector. One of the very first targets of caspase are the fluorescent proteins. So they're quenched immediately at the onset of apoptosis. And so if we co-stain with any fluorescent protein, in this case, cyan fluorescent protein, and propydium iodide, which stains the nucleus most intensely, you can see that there's really no overlap. It doesn't matter if we use cyan fluorescent protein or yellow or green. The amount of overlap between propydium iodide positive cells and fluorescent proteins is about 2 in 1,000, so very, very low. So this looks like a nice, robust measure. What we do is we follow neurons over time, and we look to see when they stop fluorescing as a marker of when they die. And following over time is a little bit tricky, but there's a lot of great software now. This is TrackMate and the ImageJ software suite from NIH. You follow the neurons, and you can show that when you track fluorescent protein emission, only about 1% of the neurons die per day. And it doesn't matter whether the slice is seizing or not. That's quite a bit, 10 times lower than the amount that we found from propydium iodide staining. So what's going on? Well, it turns out the microglial phagocytosis can become overwhelmed after brain injury. There's enough microglia in the brain to take care of neurons that die here and there, but if you have an MCA stroke, block off your middle cerebral artery, and billions of neurons die, that's a task for the mitochondria. For the microglia. So here's a serendipitous finding that AM dyes that Roger Chen developed to get dyes into the cytoplasm of cells by masking any carboxylic acid moieties, the ionization of which would prevent membrane passage. So we use this methyl group here to do that, which had an ester link to the fluorophore, and then the dye crosses and gets deesterified and stays in the cytoplasm. So it turns out that in the organotypic slice preparation, AM dyes exclusively label apoptotic neurons. They don't label live neurons shown here. So here's turbo-RFP, or red fluorescent protein. They only label cells as they die and stop expressing the fluorescent protein. They don't label astrocytes either. So a nice marker of neurons in this, apoptotic neurons in this preparation. And so if we co-label with SBFI-AM and isolectin, which labeled activated microglia, we can see how long it takes for a neuron to be engulfed by a microglia. So that involves the microglia approaching the neuron finding the neuron, recognizing that this is an apoptotic neuron, and then engulfing it. So this, in this particular case, took about 3 days for that to happen. And if we look overall, we find that neurons that are SBFI labeled, so they're definitely apoptotic. Time constant from the time of apoptosis induction, so when they first become SBFI positive, like up here, to when they disappear, time constant is 5 days, and it can be up to 2 weeks before the neurons are apoptosis, or before the phagocytosis, sorry. So seizures and activity in the slice or in the brain slows down the microglia. They're trying to sense those eat me signals and find me signals to find the apoptotic neurons. And the more active this network is, the more glutamate's being spilled, the more purines are being spilled, the harder it is for the microglia to find the dying neurons. And this is an example of microglial movements in control conditions. If you block the neural activity, the microglia become much more rapid to find their targets much more easily. If you then add ATP or glutamate to the TTX, exogenous find me signals, then the microglia become confused again and don't do their job. So the question then is, when we block neural activity, are we doing something neuroprotective, or were we just making the microglia's job easier and accelerating microglial phagocytosis? So we can take microglia out of the equation by blocking, by removing the microglia with this selective toxin clodrazone that's easy to apply to organotypic slices. And then we can repeat that experiment where we put kynurenate onto the slice. And so now the question is this neuroprotection, or is this faster phagocytosis? And so if we look at slices in which we've depleted microglia, now there's no clearance of neurons at all, and the number of propydium iodide positive neurons goes up a lot. If we look at a slice where we depleted the microglia and now we put on kynurenate, we don't get any neuroprotective effect. In fact, we get a neurotoxic effect. So we get more propydium iodide positive cells, even in the absence of any effects, on microglia. If we do the same experiment, but we're not doing it with propydium iodide now, we're using fluorescent protein expression, we see that when we deplete the microglia, there's no change because the healthy cells don't care. But if we block activity, then we get a huge wave of apoptosis. So we kill off half of the healthy developing neurons, which is consistent with this increase in propydium iodide that we saw over here. So blocking neuronal activity accelerates microglial phagocytosis. It's toxic to healthy neurons. It's not neuroprotective at all. Despite that, looked like overwhelmingly positive series of studies that I showed you earlier. So the message is that after acute brain injury, the assumptions that underlie measures of the efficacy of the intervention may not be valid. Neurons queue for a long time, waiting phagocytosis. And so the number of dying neurons that you see, particularly in a one-time shot evaluation, if you sack the animal and do some immunohistochemistry, is not attributable to the rate at which neurons die. It's also attributable to their visibility. So if you do something to increase the rate at which these dyes permeate, or the rate of clearance of the dying neurons by microglia, that may explain the change in the number of dead neurons, not the rate of neuronal death. So it's just an example of something that doesn't have anything to do with being a crummy scientist or being someone who doesn't do reproducible results. It's just the fact that the biology of brain injury is incredibly complicated. And when we study it with empirical markers, such as these modified textile dyes from the 19th century that happen to go into dying neurons, we have to be prepared that we're going to get a lot of unexpected results. And unfortunately, the same is also true for diffusion-weighted imaging, which is another extremely empirical and poorly understood but robust marker for injured brain tissue. So if we wanted to have a clinical correlate here, we could ask how many penumbral neurons are dysfunctional after misery perfusion or during misery perfusion. So in this penumbra between the DWI positive stroke and this area of misery perfusion that we might try to save with mechanical thrombectomy, we hope that all the neurons in this area are hanging on and still salvageable, but they may have already contributed to apoptosis, and they may just be undergoing a delayed clearance. And so reperfusing that area is not going to do anything at all. And in the worst case, it may even be, and this seems to be the case in trauma, that the presence of apoptotic neurons are contributing to the penumbral oligemia. So this penumbra may actually be marking neurons that are just awaiting phagocytosis, not awaiting reperfusion. So some things to consider, I think it's nice, not pleasant things to consider, but much more pleasant to consider this than the idea that, we're doing intentionally sloppy science. So let's see if I'm going to be, I might have to go off of this for a second. Oh, cheat, use my finger. There we go. Okay, so the next topic is our acute seizures in the setting of brain injury. Seizures are a robust biomarker for chronic morbidities after hypoxic ischemic injury. This is a study from Hannah Glass looking at the frequency of seizures and outcome. So if you had neonatal hypoxic ischemic encephalopathy, so asphyxiated birth, but you didn't have any neonatal seizures, nobody developed chronic epilepsy. If you just had clinical seizures only, so you weren't diagnosed by EEG, and only about a quarter of those clinical seizures turned out to be electrographic, you had reasonable incidence of epilepsy that developed over the first 6 years of life. If you had documented EEG positive seizures, so a higher frequency, there's a higher incidence of epilepsy. And if you had neonatal status epilepticus, so the highest seizure burden was associated with the most severe epilepsy that developed most rapidly. So clearly a good biomarker for chronic morbidity, but really the question is it a cause of chronic morbidity? And so there's this Telstar study, a Dutch study that came out this year in New England Journal, where they took adults who had been subject to hypoxic ischemic encephalopathy after cardiac arrest and presented with coma. And they asked, and had electrographic seizures on the EEG. If we suppress the seizures, would they have a better outcome? And they found, and their answer was no, they couldn't discern a better outcome after suppression seizures, although the actual independent variable was the treatment. It wasn't the actual suppression of seizures, which occurred about 56% of the time. And the treatment wasn't an anticonvulsant. It was an anticonvulsant plus anesthesia. So they got an infusion of midazolam, propofol, or pentobarb. And then finally, they had an enormous amount of mortality, as you'd expect. So these are people comatose after cardiac arrest. But if you look at the people who woke up, there was a better outcome among the survivors, about a 25% increase. So that would be a number needed to treat at 4. So it's possible that there's an impact of seizure control in this negative study. But I think this study also shows that we need a much more effective treatment for seizures after brain injury before we can answer the question, are seizures making outcomes worse? So if we want to come up with a better treatment, we first need to think about the pathophysiology. And I'm just going to show you here the canonical pathophysiology of acute seizures after brain injury. So a variety of bad things: mitochondrial dysfunction, there's plain old ischemia, excess ATP consumption during seizures can lead to cellular ATP depletion, which causes sodium and calcium pumps to fail, membrane depolarization from the sodium pump failure, glutamate release from the calcium pump failure, exogenous or extraneous action potentials, and seizures. So I think we would all, if we were filling out a boards test, we'd all say, yeah, that's a great pathophysiology for acute seizures after brain injury. But the problem is the timing is off, right? This should all happen in the course of seconds to minutes. It doesn't take very long to deplete cellular ATP. But we can have a transient ischemic attack that lasts for hours, in which I might be densely hemiparetic, so those neurons are not working, but there's no seizures. So cellular depletion to the point of complete functional failure doesn't lead to seizure. So where you would expect it would with this pathophysiology. And the second thing is seizures in the setting of hypoxic ischemic encephalopathy don't take minutes or tens of minutes, they take many hours. So if you look at the Telstar study we were just looking at on the previous slide, most common onset of seizures was between 24 and 48 hours. So far away from the actual hypoxic ischemic event. This is a little bit better worked out in neonatal brain injuries, where the vast majority of brain injuries are caused by acute brain injury. So hypoxic ischemic encephalopathy, stroke, hemorrhage, or infection. So acute brain damage in red, and a variety of genetic etiologies in blue that we're not going to focus on today. So if we look at acute brain injury in babies, we can see that people have worked out the pathophysiology nicely, so we know when the baby was born. And these are 15 patients, raster diagrams of seizures, so each black mark is a seizure, and an individual row is an individual patient. And over the first 120 hours of life, we can see that seizures really don't occur much in the first 12 hours, crescendo at about 24 hours, and then taper off thereafter. All these babies were treated with phenobarbital, and these authors considered phenobarbital treatment to be so ineffective that they could study the natural history of neonatal seizures, even though these patients were treated. This is a similar study from Children's Hospital of Philadelphia, where they used cooling. So these babies are cool, these babies weren't. This is a slightly different display, but it's the same time course, 120 hours. The thin bars are where there was EEG monitoring. The thicker bars are where there were a lot of seizures. The really heavy bars are status epilepticus. And this light area is where they were cooled, and this is where they were rewarmed. And so you can see that cooling, again, these were all treated with anticonvulsants also. Maybe the rewarming triggered a few extra seizures, but overall same crescendo, decrescendo pattern that if we were to draw a freehand would probably look at like this. So very few seizures in the first few hours of life. I'm just going to stop for a second and asking, why would phenobarbital work so poorly? This is the first paper I published when I left Istvan's lab. And I had spent a residency treating patients with phenobarbital with neonatal seizures and not seen much effect. And Ben-Ari had just come out with a paper saying that giant depolarizing potentials in the hippocampus were driven by GABAergic receptor activation. And so I thought, well, maybe if GABA is excitatory, then drugs like phenobarbital, which are allosteric modulators of the GABA receptor and prolong the open probability of that channel. And if GABA was depolarizing and excitatory, then that could be an explanation for barbiturate failure. So I set up this experiment where we increase the chloride concentration in the pyramidal cell a little bit, so that when a GABA operated channel opened, instead of negatively charged chloride flowing into the neuron, it would actually flow out of the neuron. The neuron would lose negative charge and become depolarized. Would that trigger action potentials on what would happen to the action of barbiturates in that situation? So we stimulated an interneuron input and activated GABA receptors, and instead of an inhibitory response, an inhibitory postsynaptic potential, we got an excitatory postsynaptic potential with an action potential. So enough chloride left the cell to trigger an action potential. When we put on pentobarbital, we open the GABA channels longer. We just prolonged the excitatory response and the number of action potentials. So this is a nice example of the plasticity of the GABA receptor. So there's lots of ways that GABA receptors can change, phosphorylation and subunit changes that I think Richard Olson has described over the years. But there's this other plasticity also, which is the reversal potential. So if the neuron's resting membrane potential is minus 65 millivolts, the GABA reversal potential could be all over the place. And by GABA reversal potential, or E-chloride, which we'll use a lot in the next few slides, I mean the value to which an open GABA receptor, currents with the open GABA receptor, are going to drive the membrane potential. So if the GABA currents drive the membrane potential to minus 80 millivolts, that's going to hyperpolarize the cell. It's going to shunt excitatory currents. inactivates open sodium channels. So a lot of things that are inhibitory, obviously, as you get down into this territory. But as you get up higher, you get reversal potentials of -50, then you're activating low threshold calcium channels, you're removing the magnesium block of the NMDA receptor, and if you go up higher, you activate sodium channels like we did up here to trigger the action potentials, or even high threshold calcium channels. So the GABA reversal potential, which can vary over all this area, is critical to the end result of GABA A receptor activation. It doesn't have to be inhibitory. So I really wanted to know how is E chloride determined? How is the GABA reversal potential determined? Of course, this is the Nernst equation, and this is all a bunch of constants. And the thing that really matters is the ratio of intracellular chloride to extracellular chloride. So really the question becomes, what sets intracellular chloride in neurons? And how does that change during acute brain injury? And on this slide, I'll just show you what I had mentioned before, that during the course of apoptosis, the membrane permeability continues to increase. So if we look at just the resting membrane potential, and this is in high potassium culture media, fluorescent positive neurons, so healthy neurons expressing fluorescent proteins, have a resting membrane potential about -55. Neurons that have recently started to take up AM dye, so these are neurons early in apoptosis, have a terrible resting membrane potential, -25 millivolts. So they clearly have a leaky membrane. They're emitting sodium, and the farther in apoptosis they go, the more sodium they emit. So if we look at SBFI AM positive neurons, SBFI is actually a ratiometric sodium dye. That's why the reason we got into these experiments. So the SBFI positive neurons that don't stain for propylium iodide have an elevated sodium, mildly elevated sodium, and by the time they stain for propylium iodide, they have a markedly elevated sodium. This isn't due to failure of mitochondria, because whatever potential, whatever sodium concentration we find in a neuron, if we block oxidative phosphorylation, we get a robust increase in the sodium. So the neurons are pumping sodium out. We can see that also by blocking sodium potassium ATPase. They're making ATP, and they're using it to power sodium potassium ATPase to pump sodium out, because if we block it, sodium potassium ATPase with Wabane, the sodium concentration doubles. So they're pumping it out, but it's leaking in through these leaky membranes. Of course, we wanted to know the molecular mechanism of the membrane leak to find a small molecule to fix this problem. We looked hard at gap junctions and pannexins, which are gap junction hemichannels. So this is a organotypic hippocampal slice culture, CA1. The green are pannexin channels present before. We did another injury, which was oxygen glucose deprivation, and the red are pannexin channels that were inserted after the oxygen glucose deprivation. And if we compare neurons that had new pannexin insertion, or any pannexin insertion, to neurons that did not, we see that neurons with pannexins inserted had a much larger increase in chloride after oxygen glucose deprivation than those that remained healthy. And we can see pannexin insertion after trauma. So this is a piglet brain. The brown stains are pannexin positive neurons after trauma. This just summarizes the degree to which we see pannexin's insertions after trauma in the injured versus the contralateral. So this is the contralateral hemisphere. So normally, of course, there aren't any pannexins, and the chloride permeability in the brain is provided by cation chloride cotransporters. So there's KCC2 and NKCC1, and here we've got KCC2 stained in red and NKCC1 stained in green. Here's a hippocampal pyramidal slice at postnatal day 21, undergoing a transition from NKCC1 expression, which is dominant early in life, to KCC2 expression later. So here's an NKCC1 positive neuron, here's a KCC2 positive neuron, but they're neurons that express both KCC2 and NKCC1. And that's a little bit hard to understand, because KCC2 is an very high velocity transporter. It moves many millimolar of chloride per second. And so it is limited really by the potassium reversal potential, which is what it uses to power that chloride transport. And under physiological conditions, the intracellular chloride should be about 5 millimolar. NKCC1, though, in addition to KCl transport, also transports a sodium. And so the sodium gradient really powers NKCC1 and pushes it so that it reaches equilibrium at chloride concentrations of about a little bit less than 60 millimolar. So if we have these neurons that have both NKCC1 and KCC2, it seems like they'd be at odds. So KCC2 would be trying to export chloride and NKCC1 would be trying to bring chloride in. This isn't a very energetically feasible situation. And the solution to that problem is that there is not one cytoplasmic chloride concentration. The cytoplasm is divided up into tons of microdomains. So if we use a chloride-sensitive dye, in this case, a superchlomelion, a derivative of the chlomelion that I showed you before based on yellow and cyan fluorescent protein, we see that they're all different concentrations in the dendrites and soma of every neuron. And so a GABA synapse here might have a chloride concentration of 18 millimolar on the inside, so frankly excitatory response to GABA. And a neuron located here might have a chloride concentration of 6 or 8 and have a primarily inhibitory response to GABA. This is in an interneuron, this is a pyramidal cell, so we see the same thing along the dendrite, main dendrite of a pyramidal cell. If we look in vivo, we see the same thing. So this is an interneuron that was oriented, its processes were oriented horizontally to the plane of imaging. So we didn't have to worry about a differential transmission of different wavelengths of light. And we can see the same types of microdomains in vivo. Now the idea for this is that we think that chloride is, the reason those microdomains are there are not because there's more NKCC1 or in one place and KCC2 in another, because that would lead to a lot of chloride flux from a high chloride area to a low chloride area. But these are actually, these areas are all in equilibrium with each other. And the places that have a little bit of chloride have a lot of surface negative proteins, so fixed anions in that area. And places with a lot of chloride, these orange hotspots have fewer fixed anions in that area. This isn't a neuron, this is a fibroblast, but it's a nice demonstration of, they looked at the location of 41 different cytoplasmic proteins in the cytoplasm and showed that there's obviously a great deal of heterogeneity in here. A lot of these proteins are very heavily negatively charged, like actin and tubulin, which make up the microfilaments that make axons and dendrites possible. And these are also very heterogeneously distributed in neuron processes. So we think that if you accept the idea that there are chloride microdomains, so a lot of chloride around positively charged surface positive proteins and a lot of potassium around negatively charged proteins, that you can then explain, the action of KCC2 and NKCC1 in a single neuron, where NKCC1 is located in areas where there's a lot of cytoplasmic chloride. KCC2 is located in places with less cytoplasmic chloride. And you can work out all the complicated equilibria, and it all works out nicely. So it's complicated, and a problem arises if you then just stick a pannexin pore willy-nilly into the cytoplasm. So if you stick a pannexin pore in a low chloride area, pannexin has a different equilibrium condition. It's permeable to these moieties. And you end up with even more chloride than NKCC1 wants to transport in, a lot of sodium, a little bit of potassium, because the sodium, potassium, and chloride reversal potentials all have to equal each other before a non-selective pore like this comes to equilibrium. So this is 340 millimolar of osmoles inside the cell, so the cell is going to swell when this happens from the saltwater influx. And we can show that in a bunch of ways. Chloride accumulates in neurons injury by oxygen glucose deprivation. So it's a model of stroke to stop the oxygen and glucose flux to neurons. You can see that after that, there's a lot of yellow and red neurons that have increased chloride. The same, we can see that graphically as well. So these neurons were injured by oxygen glucose deprivation, but we didn't do it long enough to quench all the fluorescent protein. Trauma, the same thing. I showed you the data from Velody from 2012 that slicing trauma causes a lot of increase in chloride. In vitro, this is an in vivo experiment using a pig, which has a nice gyroencephalic cortex. So it appears where the pig was injured with a controlled cortical impact. We can show decreased diffusion in the lesioned area. And if we look at superclamalin expressed in these layer 2 neurons by AAV viral vector, we can show that chloride increases in the intracellular area also. So after injury, chloride is increasing and causing GABA to be excitatory, causing anticonvulsants to fail. How can we stop seizures? Well, we want to keep chloride out of the neurons. And we have been harping on this for a long time. When I sat down and looked at all the papers, why are we so fixated on using bumetanide? We're not. We've used a bunch of other agents, but bumetanide is just the most consistently effective. So we can see that after oxygen glucose deprivation in control conditions, chloride goes up. If we do in the presence of bumetanide, chloride is stable, it goes down a little bit. So I talked a lot about pannexins a few slides ago. What if I use a pannexin blocker? And there's some tantalizing things available, like the antimalarial mefloquine is a great pannexin blocker at 1 micromolar. So 2 problems. First of all, here's the control increase in chloride. You know, it has a modest effect on chloride accumulation after this was oxygen glucose deprivation. But look what happens if we go back to that single neuron model of necrosis here. So we lyse a single neuron and we get injury to all the surrounding neurons. If we do that in the presence of a pannexin blocker, here we use a specific antagonist, 10-panx, we see that we wipe out the whole slice. So neurons need those pannexins to be working so that they can tolerate the ionic sort of flux that happens after rupture of a single neuron. So if we, in the presence of pannexin blockers, if we kill a single neuron, we pretty much kill the entire slice, probably through spreading depression. But there is hope there. If we, instead of using pannexins, we just remove the chloride from the bath, we get this remarkable recovery. We weren't the first lab to recognize this. I think Mark Goldberg and Dennis Troy's lab did this experiment in cultured neurons 25 years ago. But it's a really robust and thought-provoking experiment. If you remove the chloride from the in vitro environment, extracellular chloride, and you repeat this experiment, you can kill off a neuron and get almost no surrounding injury or increase in chloride to the surrounding cells. So how can we do that in vivo? We can't suck all the chloride out of the brain's extracellular space, but we can shut off the chloride influx from the blood. So this, if we go back to our plumbing analogy, this would be like shutting off the main valve to your house if your toilet's overflowing. So there's a lot of NKCC1 in the blood-brain barrier at the interface between blood vessels and astrocyte footplates. And a lot of people have shown that in the setting of transient injury, so a transient arterial occlusion with its reperfusion afterwards, that bumetanide decreases cerebral edema and improves the outcome. That's true with infarct models. It's true with trauma where the vessels stay open. It's true with seizures when the vessels stay open. This is Andre Maserati and Raman Sankar from here. It improves the outcome after neonatal HI when arteries are open, but it doesn't work if the arteries remain occluded, or if you damage the arteries so much that the NKCC1 really functionality isn't important because you've damaged the basement membrane.
Kevin, you have to be out of here in 5 minutes.
Okay, no problem. Sure. So I'll just go to the clinical trial. Janet Soul ran this trial for 15 years as her primary academic activity. So, we say Columbus is a great guy for sailing across the Atlantic without sight of land for 36 days. And Janet was blinded to the outcome of this trial for 15 years. So we took at-risk neonates who were post-hypoxic, increase in chloride. We did continuous EEG monitoring, which was innovative 15 years ago. It's not anymore. We looked for seizures, and we thought that one part of what was going on was an increase in chloride and change in GABA signaling. Everybody got treated with standard of care, which is phenobarbital. If people continued to seize, then they were randomized to either more phenobarbital, which was standard of care, or phenobarb plus bumetanide. And these are the results. This is seizure severity versus treatment arm. So control 0.1, 0.2, or 0.3 milligrams per kilo of bumetanide, which is the maximum renal dose. We were scared to give any more than that. And if anybody tells you that a phase 2 trial is not designed for efficacy, listen to them. I didn't really believe it. There's 43 patients. That's way more rats than I ever enrolled in a study. But even in this condition, 6 of the 8 least severely affected babies were randomized to placebo, and 12 of the 13 most severely seizing babies were randomized to bumetanide. It turned out to be not a bad thing. So this is the range, depending on the subgroup here, shown as bar plots. So the most severely seizing babies were in the 0.3 milligram per kilo, so the maximum dose. But those are the babies that had the biggest effect. So if we look at the difference before versus after bumetanide, we see that the most severely seizing babies did the best at the highest dose of bumetanide. And that's shown for individual patients here. So this is the entire study, every patient. Fortunately, there was one baby with phenobarbital right here who had a fair number of seizures and didn't respond very well, which a lot of people have shown. This is Renee Shellhouse's retrospective study showing that the more seizures you have, the poorer is your response to phenobarbital. Whereas patients tend to do better with bumetanide. We didn't hit the top of the dose response curve. In our next trial, we'll have to look at that in more detail. So here's the conclusions. So why do seizures happen? Membrane permeability increases, depolarizing neurons and emitting chloride. Why are they delayed for several hours? That's the time for the membrane permeability to deteriorate, time for chloride to accumulate. Why do they stop after 3 to 5 days? A lot of those neurons have died, lost their connections, or been phagocytosed. Why don't they respond to therapy? Because AEDs work for the most part by increasing the GABA conductance, but GABA currents are depolarized and excitatory in these conditions. So we need to starve the neurons of chloride, most feasibly at the blood-brain barrier. There are a lot of transporters at the blood-brain barrier, not just NKCC1. So future directions, in addition to figuring out when is the best time to give bumetanide, I would favor giving it in the delivery room to prevent chloride accumulation. We need to find and block all these other chloride leaks and think about the role of extracellular matrix injury in chloride accumulation. So let me just get rid of this. Go back. It looks like we're not going to have time for any other bubbles here. Sorry about that, but happy to answer any questions if